Let's see why is the name sandwich. Initially in this experiment, we'll coat antibody at the base, which is a capture antibody. We coat a capture antibody at the bottom, which will bind to one of the epitopes of the antigen, and then the other epitopes of the antigen will be used by the HRP or enzyme tagged antibody, which is at the top. So this arrangement looks like a sandwich. That's why this experiment is called as sandwich ELISA. Let's see what are the things we use in this experiment. Capture antibody, and this is coating buffer with which we coat this capture antibody in the wells. This is the blocking buffer that we use to block the non-specific spaces. This is the test antigen and standard antigens that we add in this experiment. PBST will be used for washing purpose. This is the HRP conjugated antibody which will be added on top. So after adding this, it will make a sandwich. This is the TMB hydrogen peroxide substrate, which will be converted to a chromogenic substance after the reaction with the HRP. And this is the stopping solution, which will stop the reaction, after which we can read the OD in the ELISA reader. We will be using a 96 well micro titer plate like this, a flat bottom plate, and we have to add the capture antibody like that, and we have to incubate for like overnight with the capture antibody, and eventually it will coat at the bottom of the plate like this. In between, we have to wash away in the morning. After overnight incubation, we have to wash away these plates so that we remove the excess or unbound antibodies. Followed by that, we have to go for blocking, which means leaving the active sites of the antibody, we have to block the remaining non-specific surfaces like this. So this avoids the non-specific binding of the antigens. Okay, followed by that, we have to go for washing for 3 times, and followed by this, as you can see, the antibodies, the other places are blocked, only the antibodies active sites are like open to get the antigens. And in this picture, you can see that we have added antigens which might be specific to these antibodies. If it is specific to these antibodies, the antigens will just bind onto the antibodies. And again, you have to do a washing step here, which will remove the unbound antigens. So once the antigens are bound to the antibodies, the next step is to add the enzyme linked antibodies. Mostly we are using HRP conjugated, horse radish peroxidase conjugated antibodies. So that will be added to this plate, and this HRP conjugated antibody will be specific to the antigen that we have added. So once the antigen is bound to the capture antibody, the enzyme linked antibody will be bound to the other epitope of the antigen. So again, we have to add wash to remove the unbound antibodies, and then we are adding the substrate TMB tetramethylbenzidine combined with H2O2. As you know that we have added horse radish peroxidase. That peroxidase can break down this H2O2 into H2O and nascent oxygen. This oxygen will combine with this TMB tetramethylbenzidine to give a blue color solution. So this is kind of a chromogenic reaction. So this chromogenic test. So once you have this color, then you can stop the reaction by adding a stop solution, and then you can go for a reading. So remember, the intensity of this color developed is directly proportional to the presence of antibodies, which is directly proportional to the presence of antigens. So this is actually measuring the concentration of the antigen or protein in the sample. Okay, with that we can get in, can just see the procedure and how it is done.